So our next speaker is Dr. Mauricio González from Universidad de Chile, and he will be talking about structure of plant-associated microbial communities in the Andean Atacama Desert. I see you changed the title a little bit, but it's the same subject. Go ahead, Mauricio.
Okay. Thanks, Rodrigo. First, I would like to thanks to the organizer for the invitation to participate in this interesting meeting and the opportunity to talk about the result obtained by our group in the Atacama Desert. The main objective of our studies is to understand the effect of plant in the structure of soil bacterial community. Our natural laboratory is a transect named Calabria Lejía transect, which is located in the slope of the Andes mountain in the Atacama Desert, close to the Atacama Sala. Here, this altitudinal transect gradient have three vegetation belts. In the upper limit is the steppe, in the middle part is puna, and in the lower part is pre-puna, which contain different plant species. In particular, 72 plant species were identified, including their soil coverage, to assess plant diversity. This is a nice work that was carried out the last 10 years by Professor Francisca Díaz, Claudio Latorre, and Rodrigo Gutiérrez. In order to assess how each plant community influence the ecological interaction of the soil bacterial community in this extreme ecosystem, we select 7 representative plant species from prepuna, puna, and steppe. And in total, we obtained 126 soil samples that were collected from the rhizosphere or bulk soil close to the each plant. The information about this, the operational taxonomic units present in this bacterial community and their relative abundance was used to generate a co-occurrence network representing the bulk soil and rhizosphere for each one of the representative plant community in each vegetation variance. Consider that vegetation is restricted by extremely low temperature in the upper limit of this altitudinal transect, whereas at the lower limit of this transect is characterized by almost no precipitation. It's very arid regions. We have the opportunity to evaluate the environmental severity in order to generate different ecological patterns of interaction in this soil environment. Okay, so here we evaluate the ability to the stress gradient hypothesis to explain the interaction among members of soil bacterial community in this altitudinal transect. Originally proposed for plants, a stress gradient hypothesis suggests that negative interactions are more important in low stress environments, whereas positive interactions are more important in high stress environments. In our case, high stress environment correspond to prepuna and steppe regions. Well, this figure shows the co-occurrent network representing bulk soil from prepuna, puna, and steppe. In this analysis, the nodes of the network represent OTUs annotated at the film level, and the different colors of nodes represent different fields that are included here. The H represent positive H in gray or negative H in red. That represent a correlation of the abundance in a positive or negative direction. Okay. The node side finally correspond to the level of decay of each node in the network. Well, regarding the network ratio of positive negative connections, we observe that in the middle region of the transit, the ratio reach a level of 4.5 positive and negative interaction. And in the extreme of the gradient, we observe that this number decreased to 1.4 or near to 1. Therefore, our result did not support the stress gradient hypothesis because the positive negative ratio of interaction between bacterial taxa did not increase under higher stress environment of this altitudinal gradient. However, in the root zone, where the plant community appears to moderate the abiotic stress gradient, the positive negative ratio of the rhizosphera increase compared to the bulk soil. For example, at pre-Puna, the positive negative ratio increased from 1.4 to 3.2. Also, at the step, the positive negative ratio was increased, suggesting that positive or negative interaction may be context dependent. We believe that this could be a strategy that promotes or helps to soil bacterial community growing in this extreme environment when bacteria are associated with the plant root system. Consistently, this strategy appears less apparent in the Puna vegetation belt, where temperature and precipitation conditions are less stressful. In comparison to pre-Puna and steppe belts. In addition, we know that the higher vegetation coverage in Puna could provide higher nutrient contents, which likely tends to produce more positive or less negative interaction. Considering that, we evaluate this possibility using only one plant in this case to evaluate the effect of iron supplementation in the proportion of negative interaction in the community, in the bacterial community. This possibility was evaluated in a field experiment by supplementing for 48 hours the wool soil and the rhizosphere with 100 and 500 micromolar of iron sulfate. And we select Papostipa frigia, a representative plant on the steppe, in order to do this analysis. In the rhizosphere co-occurrence network of this plant, the positive and negative interaction increased from 2.4 to 3.2, as we observe for plant community in the similar regions. But after iron supplementation, we observe that the ratio increased in the plant after iron supplementation, but also increased in the bulk soil. In the bulk soil, we found the major change in this value. And the magnitude of the positive connection, positive and negative connection, changed from 2.4 to 6.8. This result suggests that iron supplementation reduces antagonistic interaction among members of bacterial community. And it suggests also that plant buffers the effect of iron availability at the root system. This is our data. Thanks for your attention. Sorry. I would like to thank the students and collaborators that has been participate in this scientific work. Thanks.
Thank you very much, Mauricio.